Good evening to everybody. I'm Ann Berner, your host for the Bird Study Group Speakers Series. And I'm really delighted to have somebody with us who's going to talk about her research project, which sounds very interesting. As usual, I would ask you to put any questions that you have in the chat. And Thilina is going to talk to us first and do her presentation, and then we'll go back around to answering the questions that are in the chat. As always, I like to remind everybody that I'm open to suggestions of topics you'd like to know about and speakers. If you have a speaker in mind, please let me know. I'm always happy to have suggestions. So if you've got any suggestions, I'll probably remind you again at the end because I just want to let you know that we're trying to have speakers on topics that are of interest to everybody. But before we get started, I'm going to offer a land acknowledgement. Just a second here. I'm just going to let in a few more people and then we can get started with that. Okay, those folks are in, so we're good to go. In the spirit of respect, reciprocity, and truth, we honor and acknowledge the traditional Treaty 7 territory and oral practices of the Blackfoot Confederacy, the Siksika, Kainai, Piikani, Stoney Nakoda Nations, Chiniki, Bears Paw, Goodstoney, and Tsuut'ina Nation. We acknowledge that this territory is home to the Métis government of the Métis Nation within Alberta Districts 5 and 6. And finally, we acknowledge all nations, Indigenous and non, who live, work, and play on this land and who honor and celebrate this territory. So I'm really pleased to introduce to you, and Thilina, I'm probably going to mess up your last name, so I hope I don't, but Thilina Maddegamgoda, did I get that right? Okay, so Thilina is going to talk with us about population genetics of aerial insectivores. She's got a beautiful picture of a swallow there. So Thilina, I'll let you just jump right in.
Thank you. I'll share my screen. Okay, can you all see my screen?
Yes.
Okay. So yeah, as I mentioned, I'm Thilina and I'm working as a research assistant in the Bergland. And this is about my master's project. Oops. It's about landscape genetics of declining North American aerial insectivores. So as you all know, aerial insectivores are a group of birds that feed on flying insects, usually swallows, swifts, nightjars are commonly known as aerial insectivores. So you might all know during the last few decades, aerial insectivores experienced rapid declining compared to the other passerine species. This is report from 2019 State of Canada Birds. But in the most recent report, we see aerial insectivores are not like now grassland birds are declining rapidly compared to the aerial insectivores, but still aerial insectivore experience decline. So that lead to general public, ornithologists, and researchers why these birds are declining. Is there any pattern for decline, and how do we conserve them? So that lead to different research published recently for evidence of aerial insectivore declining in North America, and what is the how, and there is a roadmap to conserve these aerial insectivores, and then is there any geographic pattern for decline, and what they found. There are several reasons for declining. They did not find, or this is the exact one reason, but there are several reasons like habitat loss, habitat degradation due to the anthropogenic modification and agriculture, and climate change, and also loss of insects due to agriculture. And when it's coming to the proportion of decline, varies by region and species. As you all can see, Let me find my laser point. Here, the sand martins here. Um, European people say this is sand martin, but in North America, we say these bank swallows, they experience here, you can see rapid decline compared to other species like tree swallows or purple martin. And also when it come into the declining by region, northeastern part of North America experience rapid decline compared to the other parts of North America. And then that leads to our curiosity. Are there any population genetic differences among these different populations? And are there any barriers to the gene flow? And then is there any genetically isolated population that needs to be prioritization in conservation? Because conservation is always costly. So if there is any genetic different population, we have to prioritize them in the case of conservation. So let's talk about gene flow. Think these are each color of the different bird represent different genetic makeup. So when there is no barriers, birds can intermix with each other so they can breed, and then the genetic diversity is high. But in the case of presence of a barrier, what will happen? Gene flow is restricted, so the populations cannot mix with each other anymore. So over time, what will happen? These isolated population evolve in isolation, and they end up having different genetic makeup. So in the case of any sudden event or any random event, what will happen if there is um, uh, no individuals that can survive in that event, the whole population can be wiped out. So what are these different barriers? There can be historical barriers, contemporary barriers, physical barriers, and behavioral barriers, and I will go through each of them separately. So talking about the historical barriers, you might all know North America was covered by different ice sheets during last glacial maxima. So what will happen when there is ice sheets? These populations were resided in ice-free refugia, ice-free habitat called as ice-free refugia. So then they evolve in isolation, and these different habitats have slightly different selective pressures. So they undergo different process, and their genetic makeup can be different over time due to their genetic drift. And then once this ice sheet melts, what will happen? They will recolonize to either their original habitat or they will migrate to new habitats. But even though in present they mix with each other, but still when we do studies, we find still the effect of last glacial maxima to this date. Another barrier is a physical barrier. So physical barriers can be mountain range or large bodies of water. So for a good example, Rocky Mountains in the west is a huge, largely non-physical barrier for many species of plants, insects, and birds. And there can be large bodies of water and fragmented habitats, which also can act as physical barriers. And when it's come to behavioral barriers, there could be sexual selection, like the tails in peacock, and then different dancers in like fruit flies that can act as sexual selection, which can also be a physical behavioral barrier, and also migratory divide. So when it's coming to migration, you all know what is migration is. There is breeding birds, and then they go to their overwintering sites in the fall and then overwinter in one location, and then they come back to their breeding grounds. Usually, migration is phenomena known as to enhance the gene flow, because migration is always mixing. However, when it's coming to flyways, let's say there is a migratory divide. So that means the breeding birds coexist together, but then they choose different paths to go to their overwintering sites. So based on the path they choose, they cannot mix with each other. So they won't mix with each other. If there is migratory divide exists, that is can also act as a migratory barrier to gene flow. So then that lead to our research question. We picked three long distance migratory birds: barn swallows, bank swallows, and tree swallows. Barn swallows and bank swallows are distributed a whole world, but as our study species, we picked the North American subspecies, barn swallow, Hirundo rustica erythrogaster, and the bank swallow, Riparia, Riparia, Riparia. So now on, if you see the figures at the top and the alpha code, BARS is barn swallow and BANS is bank swallow. And these two figures I will use throughout the presentation. And for tree swallows, I'm using the alpha code trees, TRES, and this figure. So talking about their habitats and nesting, you might all seen this. Barn swallows use the cup-shaped nests, and they are considered as special concern according to COSWIC 2021. And bank swallows considered as threatened in Canada, and tree swallows not currently at risk, but they were also considered as species at risk. All of these three birds were long distance migrant, and they largely decline in the eastern part of North America, as I mentioned earlier. However, globally, they are least concerned. And also talking about these migratory pathways of these birds, Hobson et al. Found that there is a migratory divide for barn swallows, which is birds in the eastern part of North America. They travel relatively longer distance all the way to South America compared to the western and central population. They travel relatively shorter distance toward the southern part of North America. They found that using geolocators as well as isotope analysis. And talking about bank swallows, there is no published research about the migratory pathways. However, I took a screenshot of the Motus wildlife tracking data, and there is, similarly to barn swallows, there is also west of the Rocky population. They travel through the west, but the birds from Yukon as well as the central population, even though the Yukon birds are towards the west of the North America, they tend to go over the east of the Rockies. They don't seem to fly west of the Rockies. And same as barn swallow, bank swallows, eastern populations of bank swallows also travel all the way to South America. And then for tree swallows, using geolocators, they found three, again, similar to the other two species, three main flyways. However, you can see there is some birds are intermixing here and there. So that leads to our research question. Is there any population genetic structure of these barn, bank, and tree swallows across North America and across these migratory divides? And if there is any structure, what could be the potential influence on gene flow? And we also wanted to provide insight into conservation prioritization. So what we did, we collected samples across North America. We were able to collect migratory samples and overwintering samples for barn swallows, but not for the other two species. So if there is, for Saskatchewan, Manitoba, and Colorado, we have star marks for barn swallow, which means they have both breeding and migratory birds. And for the other two species, we have only the breeding birds. And here, for the make it easier, I color coded all the western samples in shades of red and central samples in shades of pink, purple, and then eastern samples in blue. So this is what we do when we collect samples. We catch the birds using mist nets. These videos and photos are from the other fields that we did for the other birds. So what we do, we set up mist nets like this, and then once we catch the bird, we carefully extract the bird, and then we collect about 30 microliters of blood from brachial vein puncture, and then we make sure bird is not bleeding anymore, and then we release the bird at the captured site. And we also band the bird. And then what we do once we have the samples in the lab, we do DNA extraction based on the type of analysis we do. So for this project, I did microsatellite analysis, RADSeq, and whole genome. But for today's presentation, I will only talk about whole genome. So this is low coverage whole genome sequencing. So as the name implies, so for the sequence, there is two type of coverage, breadth of coverage and depth of coverage, as you can see in the figure. Breadth of coverage is how much of the genome we have been sequenced. So as name implies, it's the whole genome, so higher breadth of coverage. But the depth of coverage is low. That's why we say low coverage. So what we do, we have sequenced all whole genome, and then what we do, we trying to identify single nucleotide polymorphic sites. That means, so here at the figure at the bottom, you can see our DNA is made with bases G, C, T, A. And if there is any sites that is different, that is called as single nucleotide polymorphic sites. So we try to understand such, try to find that such sites, and we do computational intensive thing. Here a screenshot of what we do. We have to do a lot of coding, and then I'm not going to go through a bunch of codes, so I will skip that part, and then I will skip to the good part, results. So once we do all these filters, what we do, we visualize our data using a principal component analysis. So principal component analysis is a dimension reduction method to try to identify patterns. So here we have principal component 1 in the x-axis and principal component 2 in the y-axis. And if there are 2 points closer together, that means they are more genetically similar than the points that are further apart. So here you can see along the PC2 axis, there is blue clustered together, and how do I, okay. These blue ones clustered together, and then the pink, purple clustered together, and the red points clustered together. And then when I check the PC2 versus 3, you can see the pattern now more clearly. The blue ones, that means from the eastern part of North America, clustered together, and the central ones clustered together, and western ones. So here's the west of the Rockies, BC, Washington, and California, and then central samples, as well as the overwintering samples from Colombia clustered together, and then eastern samples clustered together. But you can also see there is 2 triangles. Those are migratory samples from Colorado. They cluster in both east and western populations. So that means they, even though there is a structure, they can, during migration, some birds can be mixing. And when it comes to bank swallows, we observe similar pattern, and it is more clear now. The western sample clustered together, and again, the central ones. If you remember the migratory pathway I mentioned for bank swallows, these Yukon birds, they go all the way east of the Rockies and go with central populations, and we can see the same pattern here. they cluster with the central ones. And then in the east, we can see the blue ones here separated along PC1 axis, and we can also see Ontario and New Brunswick also separated along the PC2 axis. And when it's come to tree swallows, pattern is less prominent than barn and bank swallows. However, if you disregard all these pink and purple ones, you can kind of see further west and further east uh, pulling out if you disregard the central ones. But it is less subtle compared to barn and bank swallows. It seems like the central population mix with both western, eastern populations of tree swallows. And then what we did, we also did modeling, species distribution modeling. So here what we did, we have current distribution in the left, and then we have distribution in the last glacial maxima in the right. And here, if there is red, means there's high suitability habitat, and if there is blue, yellow, green, that means there are low suitability habitats. So what we did, here's the current distribution, and here we have distribution, most suitability habitats during the last glacial maxima for barn, bank, and tree swallows. As you can see, for barn swallows, there are 3 patches in Beringia, southern of the states, and then in New Brunswick. And then for bank swallows, same, we can see nice big patch in Beringia, and then southern part, and then the eastern part. But for tree swallows, we only see one big patch here, and there is a small patch over here, but there's a large one in the southern states. And I also put, this is the same PCA, but I did in a 3D way. So I don't want to show the PC2 versus 3. It's just the all 3 principal components in a 3D PCA. So what does all mean? So coming back to our first objective, we wanted to know, is there any population genetic structure across North America? So for barn swallows, yes, we found across the migratory divide, there is, because with the Hobson et al. Paper, what they found, there is a migratory divide between eastern and rest of the population. So we found the same pattern, but in addition, we also found a central cluster within the western cluster. For bank swallows, then again, yes, we found population genetic differences. We found 3 main clusters, same as for barn swallows. For tree swallows, there is some structure, but it is more slight compared to barn and bank swallows. It seems to be the sum of the central population individuals, they mix with both eastern and western populations. So then what we wanted to do, we want to understand how that happened, what could be the potential influence on gene flow. So as I mentioned in the the introduction, this could be reason because these population could resided in multiple refugia during the last glacial maxima, and also the physical barriers also play, like Rocky Mountains could also play important role shaping this population structure. But not only that, but also the migratory pathways, because there is scientific evidence that says when the populations were isolated in ice-free refugia, the paths they choose to relocate also known as migratory paths that they use up to this day. So not only that we can't say, oh, just because of the historical barrier this happened, it is combination of the historical event, physical barriers, as well as migratory routes. play important role in shaping the population structure that we observe today. And then lastly, we wanted to provide insight into conservation prioritization. So I picked barn and bank swallows, which shows rapid decline in eastern part of North America. So we observe there is a eastern part of North America. The population, they cluster separately in both bank and barn swallows. And also when looking at the migratory routes, they travel longer distance and they have to travel um, more from the above the sea compared to the central and western birds. So when we have to prioritize, it is important to prioritize the birds from the east. So what is this all mean? So we found population genetic structure for both barn and bank swallows, and also observed subtle structure for tree swallows. And these all profoundly influenced by landscape features, historical barriers, as well as migratory pathways. And if we include more migratory birds and overwintering birds and more sampling coverage, we can make robust conclusion on conservation strategies. So that's all about my project. I also have a few slides on one of the lab mates that is in our lab. That is about house wren, effects of urbanization on population connectivity on northern house wrens in Calgary and Lethbridge. And I'm going to present few slides. He's just started his project, and we want your help to find samples. So I will go through his project briefly. So he's trying to, I think he's here, so he can also answer any questions if you have for his project. And his research objective is to determine the effect of urbanization on genetic structure of house wren in Calgary and Lethbridge. So what he's trying to see, the anthropogenic modifications like the newer residential development, industrial areas, and highways could affect the gene flow. Like in my project, I mostly mentioned the natural barriers to gene flow, but he's trying to do anthropogenic barriers, trying to see is there any anthropogenic barriers to gene flow of these house wrens. So he picked two study sites, Calgary and Lethbridge. Calgary, as you all know, it's larger compared to Lethbridge, and Lethbridge is a medium-sized one. And there is high populated Calgary, and Lethbridge is low population. And both sites fall under the breeding range of the house wrens. And those sites are ideal because there is anthropogenic modifications as well as natural barriers like in Calgary, there's Elbow River, and in Lethbridge, there's Old Man River, and they're all surrounded by non-urban areas as well. So what he's trying to do, he's trying to find the occurrence based on the species distribution modeling, and he's trying to calculate effect of urbanization by degree of urbanization. So he's going to pick 5 urban sites and 1 non-urban site in Calgary as a control. And in Lethbridge, since it's smaller compared to Calgary, 3 urban sites and 1 non-urban site. So here in Calgary, this is same as the other STMs. If you see red, orange ones, sites that are more suitable compared to the yellow, bluish sites. So he picked 6 sites based on these STMs: east of Calgary Airport, middle of city, northwest city, southwest and southeast, and a control in Clearwater Park. So we are planning to do field work this summer. So if you see or if you spotted any house wrens, or if you see any dead house wrens inside nest boxes, you can email us and let us know. That will be great help. So this will be a significant study because we want to see the effect of urbanization on wildlife, and this will be given information on urban barriers and population connectivity, and ultimately guide city planners. So I will send mention our emails again in the last slide. And with that, I want to thank you, everyone, my supervisor, Teresa, and all of my lab mates, and all the people who funded our research, um, and all the museums, bird banding stations, and friends for helping for this research. And thanks to all birds who donated the blood and feathers for science. And with that, I would like to end our presentations. If you have any questions, you can email us. If you see any rents, you can also email to these email addresses. Thank you.
Hi, it's Anne. Sorry about that. I was muted talking away to you. Um, I have a couple of questions. So I just want to make sure that I understand what you're saying. So not only the numbers of the birds create concern, but the like limited genetic variation is a concern as well.
Mhm.
That's right. Okay.
Yes.
And so those two things together make the eastern population of swallows more of a concern because there's less genetic variation and fewer numbers of the birds.
Yes. Yeah.
Okay. And do you have a sense of why? I mean, is that mostly due to barriers?
Like why,
Like,
especially the eastern
yeah,
ones
why?
declining? Yeah. So I looked up that one, and some studies say uh they travel longer distances compared to the other ones, so they have to undergo more risk factors.
Right, over the ocean.
Yeah, over the ocean. And then the agricultural practices and other habitat um, stuff that is happening in the east is more concern. That's what, like, the things that I found.
Okay. I just, have they been, those populations been influenced at all by the avian influenza?
I'm not sure about that. Yeah, I did not look in up to that one.
I think it's been mostly up until recently corvids and waterfowl that have been primarily impacted. I just wondered if you had any information about
Mm-hmm.
that.
Yeah, I will look that up too.
Yeah, okay. Um, and as well, just back to the your colleague's study there that you were
Mm,
describing. Uh,
yeah.
so what specifically is he looking for reports on numbers of wrens or just any kind of sightings of wrens or
Uh,
what
yeah,
is,
so we're going to do the field collection again as same. So we're going to catch the birds and collect blood samples and then do genetic studies. So similar to what I have done, but considering the effect of urbanization.
right. Okay,
Yeah, so if you see any sightings, then that will lead us to like where to find the birds.
right. Okay.
Yeah.
Um, there's a question here. Let me just go to the question. Could you explain again what is meant by genetic population structure?
Mm, okay. So maybe how could I maybe in the slide? So when we say there is a population genetic structure, so here in the first PCA, so here you can see these all eastern, the bluebirds, blue circles that cluster together. So that means there is, um, they have high genetic similarities compared to the other points that are way far over there. So if they cluster together, they are more genetically similar compared to the um, points that are farther away. Is that answer the question or?
John, does that answer your question? So you're looking at, oh, he's asking, does it refer to genetic diversity?
Oh, okay. So if there is high genetic diversity, what would so I think he might be asking the gene flow when I said the gene flow. Is that?
I'm not sure.
Hi, it's
Yeah,
John here.
yeah.
I haven't heard that term before. Population genetic structure. Like, I'm not. It's just can't quite figure out what structure means within that.
Oh,
Like, what are you referring to by structure?
okay. So when we see there is three clusters. So here, if I refer to the figure 2 in the bank swallows, when I see, say, oh, there is a population genetic structure, that means if we see any patterns. So this individual circle is one individual. And then if we see any pattern, like if there is any pattern or these points cluster together and these points cluster together in a different cluster, then we say, oh, there is a population genetic structure that we can see two main clusters. Um,
Okay, so if there was no, if they were all clustered in the same spot, then you would
mhm.
say there's no genetic structure.
Yeah, yes, that is correct. And in the tree swallows, like the points are kind of everywhere. If they are in one single point, then we can say, yes, there is no genetic structure, or else if they are like everywhere, all over the place without any geographical pattern or like any pattern at all, then we say we can't find a genetic structure.
Okay, thank you.
Mhm. Thank you.
So there's another question. Did you find any hybrid birds between the three species
Oh.
of swallows?
Yeah, that's interesting question. There is like most hybridization barn swallow studies that is happening in like European area and Asian area. But no, we did not look up to the hybridization, and I don't think we found any too. Like we specifically don't look at the any hybridization index or anything like that. And since most of the North American ones belong to one subspecies, we did not consider looking at that too.
So I just have a question around, so the last glacial maximum
Mhm,
species were present on the continent, well, and I'm assuming Europe as well, but on the
mhm,
North America at that time.
mhm.
Oh, okay.
Y-
They go all
Yeah,
the way back.
yeah, yeah. So they were resided in those refugia, and they came back once these ice shields melt.
I guess there's probably no way of determining what genetic diversity was when they first returned as the ice melted.
Uh,
Probably not. I don't know how you would ever get that, but it'd be
yeah, like if we have, yeah.
interesting to look at what the diversity would be at that time.
Mhm.
Just looking to see. So, why would Eastern populations travel further to their wintering grounds?
Mm, yeah. I don't know. Like why they we can have to ask from birds, I guess. I don't know. I think the most suitable habitats for them seems to be the South America compared to the other ones. I don't know this particular reason why they prefer to go all the way to there, um, not to the Central America or to the southern parts of the states as the western and central birds.
You'll have to add in an interview component, interview the birds. So, what are you expecting to find? I'm sorry, I missed your colleague's name in the study on the
Oh,
winter.
Milan?
Yeah.
Yeah.
But are you expecting to find that, those barriers created by human development are going to impact genetic diversity?
Yes, I think that's what he's hypothesizing, because there are similar research have been done for the effect of urbanization, and most of them show evidence effect of anthropogenic modification can influence gene flow. That's what I think his hypothesis is going to be.
And you're already seeing that, I think, with some mammal species where they've not
Mhm.
been just very small corridors that they can travel, so they don't have as much area that they can
Yeah,
go out to.
yeah.
Same sort of effect.
Hmm.
Wow. Well, it certainly gives us another insight into how much, human development has impacted these population groups.
Yeah, that would be an interesting study.
Mm-hmm. Yeah. I think that's all the questions. I don't, there's several comments about thanks for the presentation. I don't think there's any other questions. Is there anything else that either of you give your colleague a chance to jump in if he wants to?
Yeah, I'm
But
not sure if he's there.
okay, put him
I
on the
think,
spot just in case.
yeah.
But I think that's all the questions that we've got. Is there anything
Mhm.
else that you would like to add to what you were describing to the group?
Um, no, I think that's all. Thanks for inviting me. And then if you have any other questions at any point, you can also email us, me, Teresa, and if there is any REN related things, me, Lan, and any of us. Yeah.
Would you be able to put the screen back up with everybody's email
Oh,
address just to give people
yes.
maybe?
Oh, this live is stuck here. I'll stop share and share again. Mm, it's not moving.
Oh, okay.
Wait a second. Okay.
There you go.
Yes.
Okay,
Okay.
great. That's helpful. I'm just, oh, did it
But,
disappear?
oh no, why is that happening? Okay, I will leave it like this.
Yes, that's great. I'm just trying to take them down myself, actually.
Thank you.
Okay. Well, that's great. Well, thank you very much for your time and describing your project. That's fascinating. Are you thinking about moving on to a PhD level research project?
Hopefully.
And would you stay on a similar topic, looking at genetic diversity?
Yes, I would like to do genetics, and I like birds too. So, yeah, I'm not certain at this point, but definitely I will do something related to genetics and hopefully birds.
Well, it would certainly be great to hear from you again if you do move on to doing a project like that. I think that's one of the things that I think is an interesting focus is as we understand some of these more detailed aspects of the lives of these birds. I think it's just helpful to everybody. So that'd be great to hear from you
Thank you.
if you do go down that road.
Thank you so much.
Okay, well, thank you very much for joining us this evening. And
Yeah.
everybody, again, I just one last reminder that if you have any ideas of topics or speakers, please try to reach me through Nature Calgary. Always open to those ideas. Thanks, everybody, and good night.